Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Droplet-Based Whole Genome Amplification, Successfully Sequencing Minute Amount of Mycobacterium Tuberculosis DNA. My name is Sonia Hunt, and it's my pleasure to be your X-Talks moderator for today. Today's webinar will run for approximately 45 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Samplex APS, who developed the content for this presentation. Samplex APS supports the life sciences and medical research communities with microfluidics-based solutions designed to deliver high-resolution insights into cells and genomes. Samplex stands ready to help academic, corporate, and government researchers reach their goals in areas as diverse as engineered cell therapy, molecular engineering, and cell biology. They have developed unique microfluidics instruments to support molecular engineering, Xdrops, and Xdrop sort. Xdrop can change how you analyze and work with cells. Now, it's my pleasure to introduce you to your speaker for today's webinar, and she's Dr. Anzaan Dippenaar. There she is. Hello. She is a molecular biologist with a PhD from Stellenbosch University, South Africa, and has over 10 years of experience in tuberculosis research. She is currently employed as a senior researcher in the tuberculosis omics research consortium at the University of Antwerp, Belgium. Her research focuses on deciphering the dynamics of TB outbreaks over time, making use of next-generation whole genome sequence data of Mycobacterium tuberculosis isolates. She also investigates the evolution of drug resistance in MTB clinical isolates and specimens using whole genome and targeted deep sequencing data and optimizing methodologies to extract mycobacterial DNA from clinical specimens for downstream NGS applications. Now, with the introductions out of the way, it is now my pleasure to pass the mic and the controls over to Anzaan. So Anzaan, when you're ready, you may begin.
Perfect. Thank you so much, Sonia, and hello, everyone. I'm really excited to share our research on droplet-based whole genome amplification with you. Particularly its successful application in sequencing minute amounts of Mycobacterium tuberculosis DNA. Also listed on this slide, and let me just get my laser pointer. Also listed on this slide are my colleagues and co-authors who contributed significantly to the work that I'm about to present. So let's get started. Let's begin by understanding tuberculosis, a disease that has really plagued humanity for centuries. Tuberculosis is often referred to as TB and primarily affects the lungs, but can also target other parts of the body. It's caused by a bacterium called Mycobacterium tuberculosis. Here we have a closer look at the culprit, the MTB bacterium. It is a rod-shaped bacillus, and notice in this panel its distinct thick lipid-rich cell wall, which makes it particularly resilient. It also harbors a single circular chromosome with very high GC content, approximately 65%. And this contributes significantly to its unique genetic makeup and many of the challenges that we face in TB research. So here you can see that TB is still a pressing global health challenge, even in 2023, as this map depicts the global distribution of TB cases, emphasizing really the critical need for robust diagnostics and treatment strategies, even in this day and age. So despite our advancements, TB remains the deadliest infectious disease killer worldwide, second only to COVID-19 from 2020 to 2023. Particularly affected areas are the African region and Southeast Asia. But to echo the mantra from COVID-19, we're not safe until everyone is safe, and that's really why we need to tackle the issue of TB. So whole genome sequencing as a technique has been applied in the world of TB research since approximately 2010, and we've really come a long way to also reap its clinical and public health benefits. So whole genome sequencing enables us to construct detailed phylogenetic trees, such as the one that you see here. And by analyzing genetic variations, we can track the evolutionary relationships between different TB strains, aiding in surveillance and epidemiological studies. At its core, whole genome sequencing also allows us to unravel the intricate physiology and pathogenicity of NTB. So by examining its precise genetic makeup, we can gain insights into how the bacterium functions and how it causes disease. Then whole genome sequencing also plays a critical role in identifying drug resistance mutations and also minority variants within NTB populations. And this information is really vital for tailoring effective treatment regimens, as drug-resistant TB poses an even greater public health threat. And then in the realm of public health, whole genome sequencing serves as a powerful tool for outbreak detection and investigation. And by analyzing transmission networks, we can trace the spread of TB within communities and then implement targeted interventions to curb further transmission. So from research to patient care and public health initiatives, the application of NTB WGS are quite vast and transformative. But NTB being unique in many ways, as I've mentioned, performing whole genome sequencing does come with challenges. The conventional workflow for NTB whole genome sequencing is very time-consuming. And this is in part due to the slow growth rate of NTB in culture and the current sequencing technologies that we use need for large amounts of pure NTB DNA. And I put an asterisk here after the only NTB only DNA because small amounts of non-NTB contamination can be overcome bioinformatically, but for the most part, it's preferable that the DNA for sequencing consists mainly of NTB DNA. Now, if I walk you through this diagram, it all begins with the collection of a clinical specimen, typically sputum from a patient suspected to have tuberculosis. This collected sputum sample is liquefied and some of the non-NTB contaminants are then removed in this process. A strong alkaline solution is typically used, we call this milk, NaOH, used to treat the sputum before it's then inoculated into the culture state. So next, the sample undergoes NTB enrichment, often through automated liquid culture methods, such as the midget or Mycobacterium growth indicator tube system, and subsequent subculturing on then solid or liquid media further increases the NTB biomass while removing additional non-NTB contaminants. And this process of subculturing is often done in research lab settings. Now, after this enrichment steps, to ensure biosafety, the NTB culture is inactivated, typically through heat exposure. Then, following inactivation, NTB cells are lysed to release their DNA. This can be achieved enzymatically or mechanically, breaking down the cell walls and then releasing the genomic material. Note that this process for NTB is often more aggressive compared to lysis steps for other bacteria due to the very tough cell wall. The DNA is then extracted either by in-house or kit-based methods and typically undergoes some form of quality control, such as fluorometry or spectrophotometry with a qubit or a nanodrop. Some labs also do gel electrophoresis to confirm the DNA integrity as an added measure. And then finally, the DNA is prepared into sequencing libraries and subjected to next generation sequencing on platforms such as Illumina sequencers, which are really widely used for MTB whole genome sequencing. Okay. As I mentioned, MTB grows fairly slowly, and in our ideal scenario, we would bypass this time-consuming culture step altogether and extract DNA directly from a clinical specimen, or even from a clinical primary culture, avoiding the subculture step that I've mentioned and that is grayed out on this part of the slide. And this would significantly reduce the turnaround time to go from sample to sequence. Now, in order to do this, we need to find ways around using small amounts of DNA for sequencing and eventually overcoming non-NTB contamination. Currently, culture-free NTB whole genome sequencing is a promising approach. For example, one such method involves NTB DNA capture using complementary RNA probes specific for the NTB target DNA, followed then by binding to magnetic beads for isolation or fishing out of the NTB DNA. However, this approach is fairly expensive and it's also not been shown to be consistently successful in all sample types. This brings me to our hypothesis. And our hypothesis centers around the potential of whole genome amplification to accurately and efficiently amplify the high GC-rich NTB genome, thereby enabling whole genome sequencing from samples containing very small amounts of NTB DNA. And before I delve into the methods, I'd like to hear from our own audience. Over to you, Sonia, for the poll.
Thank you very much. I'm going to launch our first and only poll question for this webinar, and there you go. The question is, how familiar are you with droplet-based whole genome amplification techniques? Are you very familiar, somewhat familiar, or not familiar at all? So please go ahead and make your selection for one of the options that we have below, and then I will leave this open for another 20 seconds just to get everyone involved in this poll question, and then I'll close the polls, take a look at the results, and then we'll get right back into the presentation. Okay, it looks like that's the majority of our audience have cast their votes, so I just want to say thank you very much, everyone, for participating. I'm now going to close the polls, and let's take a look at the results. Okay, so here are the results. In our audience, 6% said very familiar, while 33% said somewhat familiar, and 61% of our audience said not familiar at all. So thank you everyone for participating. Now back to the presentation. Anzaan, you may take it away.
Great, thank you, Sonia. I see that very few people are familiar with droplet-based whole genome sequencing, so allow me to give an overview.
Okay, perfect.
Right. Our approach leveraged the innovation of Samplix Xdrop system, a droplet generator that enables precise manipulation of genetic material. So with its ability to generate picoliter-sized single or double emulsion droplets, the Xdrop system offers versatile encapsulation of biological molecules like DNA, RNA, or even cells. And the system is also compatible with the various types of growth media. So the droplets produced are quite stable, even under conditions typically found in a PCR reaction. And what you see here is an outline of the experimental workflow that we followed on a high level. So a DNA sample was diluted to various concentrations, and I'll detail that in the next slide. After that, the diluted DNA was partitioned into single emulsion droplets with a single DNA molecule per droplet using the Samplix Xdrop system. Within these droplets, multiple displacement amplification was done using a typical MDA reaction with Phi29 enzyme, enabling the efficient and uniform amplification of the encapsulated DNA within these droplets. Following amplification, the DNA is released from the droplets and then subjected to library preparation for downstream sequencing applications. Now, our pilot study was designed to evaluate the efficacy of droplet-based whole genome amplification across varying very low input amounts of NTB DNA. Here's a breakdown of our specific experimental design. We started with concentrated, purified NTB DNA obtained from a cultured NTB lineage 2 pan-susceptible isolate, ensuring that a standardized starting point for our analyses could be reached. This isolate also had existing whole genome sequence available from the same DNA extract, and this served as a control in the bioinformatic analyses that I'll present. The MTB DNA was then systematically diluted to achieve 4 different input amounts for the dMDA reactions, namely 5 picogram, which equates to roughly 1,000 copies of the MTB genome, 1 picogram equating to almost 200 copies, 0.5 picograms equating to almost 100 copies, and then 0.1 picograms nearing more or less 20 copies. Okay. The diluted NTB DNA samples were then subjected to the droplet generation, as I've explained in the previous slide, using the Samplix Xdrop system and facilitating the efficient amplification of the encapsulated DNA. Following dMDA, the DNA yield was quantified for each input amount post-droplet WGA or NDA, enabling the assessment of the amplification efficiency across the different input concentrations. The amplified DNA underwent TrueSeq PCR-free library preparation, followed by Illumina WGS on the iSeq platform. Finally, we conducted bioinformatic analyses using our group's MTB WGS analysis pipeline, MAGMA, which is known for its robustness in handling contaminants commonly encountered in clinical samples. And MAGMA uses a reference alignment approach to the H37RV reference genome to then subsequently identify genomic variants. Looking at the whole genome amplification yield and basic WGA WGS statistics as an overview in this table, I'll walk you through the key findings. So the DNA yield post WGA showed a notable increase with higher input DNA amounts ranging from 105 nanograms from the 0.1 picogram input all the way to 2,400 nanograms yield from the 5 picogram input DNA. Across all input amounts, we also observed a very high base quality, consistently exceeding 34. And this indicates that the sequence quality was of high accuracy, and also gives us confidence in the conclusions that we draw from the bioinformatic comparisons. We observed a progressive increase in the percentage of mapped reads with the increasing input DNA amounts, reaching 89% for the control sample. So remember that we use a reference mapping approach here. And this indicates that efficient mapping of the sequencing reads to the NTB genome for higher input amounts with some possible non-specific amplification in the reactions with the lower template DNA, where fewer of the reads map to the reference genome. Then when we look at the mean depth of coverage, referring to how many times each base of the NTB genome of 4.4 million bases was sequenced, it showed a similar trend with higher input DNA amounts towards the bottom, resulting in greater sequencing depth and lower input into low sequencing depth. Then in the last column, a crucial metric for assessing actionable sequencing results is the percentage of the genome covered at a minimum depth of 5x, which also demonstrated substantial improvement with increasing input DNA amounts from the lowest input of 0.1 picograms to the next input 0.5 picograms, then almost reaching a plateau from then on. Okay. So that indicates that even for the 0.5 picogram input sample, the vast majority of the genome is covered by at least 5 reads, enabling MAGMA, the bioinformatic analysis pipeline, to detect variants in almost the entire genome, but in less than half of the genome for the 0.1 picogram input sample. Our analysis of the genome-wide depth of coverage offered some valuable insights into the sequencing performance across the different input amounts of DNA and this positive control. And in these coverage plots that I present here, you can see the sequencing depth on the y-axis and the genome position on the x-axis, all 4.4 million bases. So in the control sample, in this panel, we observed relatively uniform depth of coverage across the MTB genome, with one notable exception, this DOS-R region, a known large duplication of about 350 kb present in certain MTB lineages, including this one that we worked with. So this region exhibited approximately double the coverage of the rest of the genome, consistent with previous observations. And similarly, the diluted or the dMDA samples displayed a comparable pattern of depth of coverage, also reflecting this duplication of 350 kb. Notably, the lower input amounts, these two at the top, showed greater variation in the depth of coverage, as indicated by the larger standard deviation that you observe. And the variability decreased progressively with the higher input DNA amounts, suggesting improved sequencing robustness and consistency. So it's important to note that the MTB genome contains GC-rich and repetitive regions, and this already results in fluctuations in coverage when using a reference mapping approach, even in the control sample, as you can see here. So next, we looked at whether droplet amplification could cope with MTB's high GC content of about 65%. And this plot illustrates the GC content distribution of the raw reads, the FASTQs, across the control sample in blue, and all the droplet MDA WGS samples in the red lines. And remarkably, we observed no discernible differences in the GC content among the various samples. And this uniformity in GC content suggests that droplet-based whole genome amplification maintains the integrity of the original DNA sequences without introducing significant biases in GC composition. Next, we wanted to know whether whole genome amplification WGS samples accurately reflect the genomic variants identified in the control sample at the nucleotide level. So using a pairwise comparison of the variants observed in the control sample and each of the droplet WGA samples, we could determine the false positive and false negative variants for each input amount of DNA, where false negative variants were those only identified in the control sample and thus missed in the whole genome amplified sample, and false positive variants were those that were only found in the amplified sample but not in the control, indicating possible amplification artifacts. What you can see from these Venn diagrams is that a decrease in input amount of DNA correlates with an increase in false positive as well as false negative variants. The highest input amount sample only differed from the control sample by a total of 5 variants, so 4 false positives and a single false negative variant. Whereas the lowest input amount sample had 175 false positive variants and missed 548 variants. Now, this high number of false negative variants in the 0.1 picogram input sample is explained by the low percentage of the genome with at least 5x depth of coverage that I showed in one of the previous slides. So this illustrates that if half of the genome doesn't have sequencing data, no variant calls can be made in those regions. Next, we looked into phylogenetic analysis To assess the fidelity of the whole genome amplification WGS in capturing the original strain's phylogenetic placement. So using a maximum likelihood approach with genome-wide SNPs, we constructed a phylogenetic tree incorporating representative isolates from various lineages within the Mycobacterium tuberculosis complex. You can see that the control sample, 5 picogram and 1 picogram sample, form a coherent cluster and is also positioned at a similar branch length, whereas the lower input samples have remarkably longer branch lengths, and this is due to the higher number of false positive variants that were identified in these samples, as I showed in the previous slide. So what can we conclude from these findings? We can see that it is feasible to sequence MTB DNA from as few as 100 copies of the MTB genome using droplet whole genome amplification. Insights from the detection of false positive and false negative variants suggest that this approach holds promise for drug resistance detection. In 0.5 picogram input DNA and above, albeit with caution warranted for susceptible calls due to the potential false negatives, so necessitating a bit of a conservative approach there. Then for surveillance purposes, input DNA of 1 picogram and above appears to be suitable. However, for outbreak detection, only the highest input amount of 5 picogram proves suitable, given the current variant thresholds applies to determine the similarity of strains for identifying patients within transmission clusters. Notably for the detection of minor variants, especially for heteroresistance, and I didn't go into this in detail in this talk, but based on our preliminary findings, we don't think that using the input amounts that we've tested could be suitable for this, as the amplification process could potentially skew the true population structure of the template DNA. In our next steps, we would like to expand the scope of our study to encompass more clinically relevant scenarios, as this pilot study inevitably has its limitations and we worked with purified MTB DNA. So firstly, we plan to transition from using diluted purified MTB DNA to clinical samples, such as spiked sputum, sputum sediments, or even clinical primary liquid cultures. This shift will allow us to assess the performance of this approach in a more realistic setting. Additionally, we think that it would be valuable to focus on optimizing the dMDA process in the droplets for MTB in clinical scenarios. And while we used standard dMDA in our initial experiments, we would like to explore this potential of using selective whole genome amplification. And this involves incorporating MTB-specific oligos into the dMDA reaction, enhancing the specificity of the amplification process and potentially improving the accuracy of our results, and would also allow us to use clinical samples with some level of non-MTB contamination. Lastly, we then intend to explore alternative NGS platforms beyond Illumina. While Illumina sequencing was used in our pilot study, we're quite eager to investigate the suitability of Oxford Nanopore technologies for this workflow especially. And we'd like to specifically explore the selective sequencing or read until function offered by ONT, and this could further enhance our ability to accurately sequence minute amounts of MTB DNA from a complex, more clinically relevant background. So finally, I'd like to acknowledge the funding bodies that supported this research, including the FWO, SAMRC, and NRF. Also, a big word of thanks to my colleagues and co-authors for their contribution to this work, the Samplex team for their expert guidance and excellent service, as well as for inviting me to give this talk. And lastly, also a word of thanks to the host of this webinar series, Xtalks, for providing the platform to share our findings, and to Sonia, who's been a very kind and gracious host. And of course, thank you all for listening. I'd be happy to take questions.
Okay, well, thank you very much for that very engaging presentation, Anzaan. I'm sure everybody appreciates it. And now we're going to begin the Q&A portion of the webinar. So I'll invite Anzaan to put on her webcam. And please, webinar, to our attendees here, you still have the opportunity to send in your questions right now by using the questions window, and we'll try to attend to your questions during the time we have together. So Anzaan, you can put on your webcam, and I will start with the first question here. This audience member is asking, how does droplet-based whole genome amplification compare to other amplification techniques for low-input DNA samples? What are your thoughts on that?
Right, thank you, Sonia, for that question. So dMDA has several advantages. It is highly efficient, and it allows more uniform amplification throughout the genome, where each DNA molecule is then encapsulated in a single droplet. So in that micro, nano, or pico environment, there's no competition between molecules in the amplification step. So that really increases its performance compared to traditional dMDA or traditional MDA reactions.
Okay, all right, okay, I'm going to go to the next question now. Thank you very much for answering that. The next question we have here is, this audience member is asking, what are the main advantages and limitations of using different sequencing platforms like Oxford Nanopore technologies for tuberculosis genomics? What are your thoughts on that?
Okay, so the advantage of ONT, moving to ONT, is that the instrument has a very low capital cost. The instrument is also portable, so it's very easy to move it even to the field. And here we're really thinking about decentralization. So it makes it very useful to take sequencing to the clinics, as we say. And then another advantage of the ONT platform is that the longer reads that it produces has advantages, especially for MTB with its very repetitive genome. And then one other thing that I mentioned also in the next steps is the read until or selective sequencing function that hasn't been really explored and is fairly new, but that we think will aid in taking this to the next step and enabling it in clinical samples. The disadvantages, I would say, on the higher error rates of ONT sequencing, although there has been a lot of improvement in this area. So that in combination with some of the errors in dMDA in the very low input amounts, that's something that we need to be cautious of and understand better, I think. And then another disadvantage of ONT is possibly the high per sample sequencing costs. I think that compared to something like Illumina, it's still quite expensive.
Okay, thank you for that. Let's go to the next question here. To confirm, this method hasn't been tested on clinical samples yet. Was it on Spectrum? This audience member is asking this one. Yeah.
Correct. So that's indeed our next steps, is to move towards clinical samples. We haven't yet done it on sputum or sediments or early positive cultures.
Okay, all right. Sorry, I'm here busy getting all these questions that are coming in here fast and furious. So give me 1 second here and I'll go to the next question here. Okay, so the next question that we have for you is, transitioning to clinical samples presents challenges due to their high levels of contamination, raising concerns about the efficacy of SWGA in such settings. In the event of potential limitations with the SWGA, do you have ideas for alternative strategies?
Okay, so if SWGA, that selective whole genome amplification, is not as efficient or promising as it seems, I think we'll have to come up with alternatives indeed. And one option is to improve the sample processing steps. So here we're looking into selective lysis, so exploiting like the toughness of the NTB cell wall. It can withstand a lot. So if you can apply a very specific methods to lyse almost everything else and then do a treatment, pre-treatment such as DNAs or with another enzyme or even just a washing step to get rid of the released DNA, that could really clean up your sample and enrich for the NTB already. So we're looking into methods that could enhance that. And then also, as I mentioned in one of the previous answers, is we're really interested in this selective sequencing or read-until function from ONT that could potentially help to overcome the dirty samples or contaminated samples. So I think that it is probably going to take a combination approach, because MTB is quite special, and I don't mean that in a good way. It really throws complications at the research in every possible term.
Okay, thank you for that. All right, let's go to the next question here. This audience member is asking, could this be used for RNA sequence?
So as far as I understand, RNA can efficiently be encapsulated in the droplets, and indeed could then be used for single molecule sequencing. Yes.
Okay, thank you. The next question we have for you is, do you grow the Mycobacterium in the droplets and then lyse it in the droplets?
Right. Interesting question. That is something that we've definitely considered, but we haven't done in this pilot study. So it's something that we look into, and especially, and I didn't mention this in the talk, but especially using the Xdrop sort, it's more of a dream really, but thinking of how we can stain MTB and then in these droplets select for them, and only the droplets that contain MTB to do more enrichment. But yes, that's not something that we've done yet, but really something that we should explore.
Okay, all right. Let's go to the next question here. Get them all lined up here for you. Okay, how do you ensure that only a single DNA molecule is captured in each droplet?
That's quite a technical question, and I don't know if I have if I'm technically adept to answer that. But as I understand, the droplets are very small, and in that dispersion, 95% of the material is dispersed in single droplets, and each is encapsulated by an oil emulsion, and that then ensures that there's only one molecule per droplet.
Okay, all right. Thank you for attempting to answer that. And I think they're happy with that answer too. Okay, here's the next question. This one is, what would you recommend the best lysis step for MTB?
Also quite a loaded question, and something that our group is really working on. So what people have done is heat, enzymatic, mechanical. And from the preliminary analyses that we do in our group, it seems like mechanical lysis using some kind of bead to say it very crudely, but to bash open the top cell wall of MTB seems to be the most effective. Because with too much heat or with a very long exposure, you also risk damaging the DNA that you get out, and that in turn affects the sequencing negatively. So we think that mechanical lysis is the best approach at this stage.
Okay, all right. We'll try to squeeze one more question in here for you. Can this be used for early medical diagnosis of bacterial infections in people?
Excellent question, and yes, that's the dream, right? I think we'll only know that once we can apply this in a clinical setting, but that's definitely the idea, is to detect the presence of bacterial DNA at very small quantities before even that's detectable by culture and so forth.
Okay, well, thank you very much for those answers. We have reached the end of the Q&A portion of the webinar. For those of you who are interested in hearing more from Samplex APS, you can register for their next webinar, Adoptive Cell Therapy, Exploring and Influencing Heterogeneity of TILs and CAR T cells. And I'm going to put the link for that webinar in the chat box right now. You should see it appear here, so you can go ahead and register for this next great presentation. Okay, now if we could not attend to all your questions, and there were a lot of them that came in, the team will follow up with you after this presentation. And if you have any further questions, please feel free to utilize the email address you see there on your screen, ala@simplex.com. Okay, So thank you everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive of this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Additionally, I have shared a link in the for the recording of today's event in the chat box, which you can also share with your colleagues, so they may register for the recording here as well. Actually, give me a few seconds and I will do that momentarily, put that link in the chat box. But get even more information from our presentation today by downloading the supporting material that we have available for you under the Handouts tab. And there's one there called Xdrop Fast Encapsulation of Cells and DNA for Downstream Analysis. Okay, so I encourage you to do that. Now, please join us in thanking our speaker, Anzaan, for her time here today. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at Xtalks, we thank you for joining us. Enjoy the rest of your day. Until next time, please take care and bye for now. Bye, everyone. Bye, Anzaan.
Bye. Thanks.
Bye.